Today I will talk about not about eucalyptus aqueous extract as Cristiano just did. I will talk about the antibacterial effect of eucalyptus-based nanoparticles against tomato plants pathogens. And sessile organisms plants like tomato plants are inevitably exposed to a wide range of stressful conditions, as it is the case of phytopathogen attacks. And tomato plants are affected by several important economic bacterial diseases, as it is the case of bacterial spot disease caused by Pseudomonas syringae tomato, or PST for short, bacterial spot disease caused by Xanthomonas eubsicatoria, or shell, and bacterial cancer caused by Clavibacter michiganensis, or CMM. And these diseases, all these diseases cause great economical impacts on tomato productions, and currently farmers face a lack of effective treatment options. In fact, they are mainly resumed to the use of appropriated cultural practices, as it is the case of the elimination of infected plants or seeds and the disinfection of pruning tools, since the application of copper-based bactericides is already being restricted in European Union due to their negative environmental impacts, as Cristiano commented, and the application of antibiotics is forbidden. So it becomes urgent to develop eco-friendly strategies to control crop-related bacterial diseases. And the solution to this issue might be in eucalyptus essential oils, because they have multiple biological properties, including antibacterial ones. However, due to their low stability, high volatility, and high hydrophobicity, their field application can be limited. So the main goal of my PhD was to develop eucalyptus-based nanoparticles as innovative delivery systems, and to explore their potential to be used in the sustainable control of crop-related bacterial diseases. We selected two types of nanoparticle systems to incorporate the eucalyptus essential oil: lipid nanoparticles and keratin nanoparticles, because they are easily produced in aqueous dispersions without using organic solvents, and because they also have typically high loading capacities of the active ingredient, and they confer a great stability of the active ingredient during storage. But in the end, which is the main difference between these two systems? Due to their lipidic nature, lipid nanoparticles have a higher affinity to the essential oil, so they will confer a slower release of the essential oil, while keratin nanoparticles, due to their aqueous nature, have a lower affinity to the essential oil, so they will confer a faster release of the essential oil. Regarding the synthesis of lipid nanoparticles, basically what we do is to add an aqueous phase to a lipid phase where the eucalyptus essential oil is comprised. And after a high-speed homogenization, the resultant emulsion is cooled on ice in order to allow nanoparticle formation. And the resultant nanoparticles consist in a lipid matrix in which the eucalyptus essential oil is comprised, which is surrounded by an aqueous phase. Regarding the synthesis of keratin nanoparticles, basically what we do is we start by extracting keratin from human hair, and after obtaining the lyophilized keratin, we prepare a keratin solution to which the eucalyptus essential oil is added. And after several cycles of ultrasonication on ice, we obtain our nanoparticles, which consist in a keratin matrix in which the eucalyptus essential oil is entrapped. After synthesis, both nanoformulations were characterized in terms of different physicochemical parameters, and both formulations showed good properties. So we could continue with our study, and we evaluated their antibacterial activities. And to do so, in a microplate, we exposed the 3 different pathogens to different concentrations of both nanoformulations and also of the free essential oil in its non-encapsulated form. And the microplate was incubated in a microplate reader for 48 hours with hourly readings of the absorbance at 600 nanometers. And the results showed nothing. The results, the growth of all situations, including the control situations, was inhibited. We tried different approaches with agitation, without agitation, and the results were all like this. Nothing grew. So we started to hypothesize that the growth of bacteria in control conditions could be inhibited by volatile compounds released by the neighboring bacteria that were combined with these treatments. So, to explore this hypothesis, we repeated the experiment, but instead of doing that in a microplate, we did it in Eppendorf tubes. And instead of evaluating the growth curves, we quantified the number of colony-forming units per milliliter. And the results show at least some growth in control conditions, and also results showed that for shale, all treatments had similar bactericidal effects, while for CMEM, the bactericidal activity of keratin nanoparticles was nearly 50% stronger than the corresponding concentration of the eucalyptus essential oil. Also, it was possible to verify that lipid nanoparticles only had bactericidal properties against shale. To further explore the phenomena of the volatile compounds that were being released during the combination of the nanoparticles with the phytopathogens, we did an additional experiment where in a microplate, in one side of the microplate, we combined the treatments, we combined each treatment with each pathogen or with deionized water as control. And on the other side of the microplate, we put each pathogen growing under control conditions at different distances from the VOC source, being 0 the closest one to the VOC source and 3 the farthest one. Then this microplate was incubated in a microplate reader with hourly readings of the absorbance at 600 nanometers. And we obtained a lot of results. Basically, what we have here in the left side of the slide, we have here the combination of each pathogen with the eucalyptus essential oil in green, lipid nanoparticles in yellow, and keratin nanoparticles in blue. While here on the right side of the slide, we have the individual effects of eucalyptus essential oil, lipid nanoparticles, and keratin nanoparticles on the growth of PST in blue, shale in pink, and Clavibacter in green. Also, you can see here a control bar, which refers to the bacteria growing under control conditions without the influence of volatile compounds. And the results showed regarding the effects of the volatile compounds released by the individual treatments, the results show that each treatment individually released volatile compounds that differentially inhibited the growth of the three pathogens, being this effect more stronger or be this effect stronger when bacteria were growing closer to the VOC source. And also this effect was more pronounced when keratin nanoparticles were present in the media. Regarding the effects of the volatile compounds, single compounds can have different effects when they are in mixture. Usually, when combined, compounds can have a synergistic effect that when alone they don't have. Of course, that here in eucalyptus essential oil, the main compound is eucalyptol, but we have also other compounds like alpha and beta pinene that also have appreciate amounts that can enhance the biological properties. And for that reason, I believe that using the combination instead of the individual compounds can be an advantage.
Do you have any idea about how this compound initially the bacteria grows, or how they are perceived by bacteria?
I didn't do any experiments on that yet, but we intend to do flow cytometry in order to know the impact of these compounds at the membrane level, because we believe that it could interfere with membrane permeability. Thank you.